'cause in the end, for you and me, radioactivity.
Okay, so what we just did in uh, this last topic is we talked about the damage that we get to water molecules and to DNA. We're going to keep moving up the ladder. So our next step is going to be looking at what happens to the DNA itself, what happens to the chromosomes. The next step will be what happens to the cells, eventually what happens to the tissues, and we'll look at it from building up to the entire organism. In fact, we'll look at what happens to a whole human when exposed to ionizing radiation. So we're right now at the stage of looking at the DNA, and again, I think this puts this in context. This is one of the better diagrams I've been able to find, done by Noelle Metting, an old friend of mine. What she did is said, look, you can have excitation, which causes light and heat. Nothing's going to happen here. But if the ionization happens in through free radical formation or through direct action, it can be fixed. You can have ions that recombine. As we said, there are all those free radical scavengers in our cells. If the DNA damage is damaged, we can have DNA repair. We'll talk extensively about DNA repair in here. But if the damage is so extensive, we can get DNA mutations that occur. Mutations in somatic cells can, but do not necessarily lead to cancer. Mutations in germline cells can, but do not necessarily lead to heritable effects. DNA damage, if it is of sufficient quantity, can lead to cell death. If this cell death is sufficient, if there are enough cells that die, we can get acute radiation toxicities. We can get late tissue toxicities, as we see clinically, and we can get developmental effects in the fetus. So this sort of gives the panoply of everything you could see from radiation injury, kind of in its worst case scenario. So I keep saying the most important target is nuclear DNA. Why is the mitochondrial DNA not so important? Mitochondria do have DNA, but the usual idea is that each mitochondrion has about 10 or 20 copies of its mitochondrial DNA, and to cause damage to all those copies is difficult. And then if you think about it from a cellular perspective, most cells have 400, 500 mitochondria per cell. So damage one of them, damage 5 of them, it's not going to really matter that much. So for that reason, nuclear DNA, which is present most of the time in a 2N form with 2 copies of most genes, except for those on X and Y chromosomes, that's a much easier target to cause damage than with mitochondrial DNA. And it's this damage that's going to lead to killing, mutation, and carcinogenesis. As I said, base damage is the most common, but base damage is going to be very easily repaired. We're going to look at base excision repair. It occurs quickly, easily, normally is not a problem for the cell. Single strand breaks are also easily repaired. They're repaired by a pathway that is a component of the base damage pathway, base excision repair pathway. Double strand breaks are going to become, for us, the most important lesion because they're very common and they almost always involve mutations, and we'll talk about why in a moment. And then multiply damaged sites are also very important. They're frequently called MDS. They might be called complex lesions, and we'll talk about that about that nomenclature. On your boards, I think it's more commonly called complex damage, although multiply damaged sites is how Hall refers to it. In Hall's textbook, he says that high LAT radiation produces only high LAT radiation produces multiply damaged sites. I'll say in a moment, but I'll say it now too. I don't think this is true. There are data, it's very uncommon, but even a single electron track can produce a multiply damaged site. It's rare, but it does happen. Are there other kinds of damage in the cell? Sure. I mean, there's going to be lipid peroxidation. There's going to be membrane damage. You know, at really whopping doses, we see these things. We see these things taking effect. So it's not that, you know, all we care about is nuclear DNA. But at the end of the day, having nuclear DNA, thinking about nuclear DNA as the major target is what helps us understand the biggest consequences for radiation effects. And in fact, all the models of radiation damage are based on nuclear DNA. The doses we need to do for membrane damage are so much higher than the doses we need to do for nuclear DNA damage that nuclear DNA damage becomes more important for us clinically. So what are the things we might see? We might see that there's a change of a base, loss of a base, hydrogen bond breakage. These are all going to be base damage. We might see a single strand break, double strand break, and we already said double strand break is two single strand breaks close to each other. We might see cross-linking. Cross-linking is the least common of all these. And the usual damage in the cell is base damage is greater than single strand breaks is greater than double strand breaks in frequency. And here's our old chemical diagram here. And you can imagine if we get a damage, an ionization event here on the base, we're going to get base damage. If we get here, we're going to get single strand break or a double strand break, depending on how much is broken. And we can get a single strand break even if the sugar residue is damaged. So there are many options for what can happen here. And as I said, there can be cross-linking as well, but that is not as common as the other forms of damage that we see. Okay, here's a very old diagram, but it's going to make my point well, and so therefore that's why I'm going to use it. Why are single strand breaks easy to repair and double strand breaks are so damaging? That's the question. So here's the reason why single strand breaks are not so bad. You get a single strand break, the DNA stays together. Send in repair machinery, fix this, and it's all done. The only thing that might inhibit this is if there's peroxidation that takes place as well. Sometimes peroxidation will inhibit repair machinery, but this is relatively uncommon. So the most common thing that happens, single strand break occurs, DNA holds together, and there we go. Problem with double strand breaks is we get two breaks that take place. There's nothing to hold the DNA together. It falls apart. So there are going to be two events that are going to take place here that are going to create problems for us with DNA, with double strand breaks. Here's again the single strand break. Even if it involves base damage, the cell knows you got to put an A in here. This break, let's say that you have a break that takes place like this is a double strand break still, but it's about 3 bases apart. Two possible consequences. Number one, there are DNases all over the cell. As these two fall apart, bases get chewed up by the DNases in the cell. They get joined together again, and we lost genetic material. If it happens in repeated DNA, it might not cause a big deal. But if it happens in a coding region, we're going to lose a protein function. We might even lose the entire protein because it caused an error in it caused a stop codon to appear in the wrong place. So this is dangerous because of the chewing up of the ends with DNases. And many sequencing of these lesions have shown us that in fact there are missing bases around these breaks. The second consequence is one we're going to talk about when we talk about chromosomal injury in just a few moments. This change involves a break, and the DNA strands don't necessarily join up together with each other. They join up to other DNA fragments within the cell because there's nothing holding these two together. Unlike the single strand break where these guys are held together, these now can fall apart and they can join up with something else instead of with each other, and then you're going to have an even greater mutation on your hands. Now, I don't want you to believe that all double strand breaks always lead to error. There is one type of repair that is called error-free repair. It is called homologous recombination. We will talk about it in detail when we talk about homologous recombination repair and DNA repair. But what happens here is a double-strand break uses a duplicate of its chromosome to repair itself. When do we have a duplicate of a chromosome? Late S, G2 phase of the cell cycle. So if the break occurs, if the damage occurs in late S or G2 phase, when the DNA has already been duplicated, then we can find homologous recombination can work. And this allows us to copy the other chromosome and return back the right sequence. What's the most common phase of the cell cycle in mammalian cells? Well, most of the cells in our body are G0 phase. Most of the cells in a tumor, if they're cycling, are in G1 phase. So late S G2 phase is among the least common of the phases of the cell cycle, so homologous recombination is not going to occur with great frequency inside the cells. But it does occur, it is error-free. The most common type of repair that's going to take place is going to be in G0, G1, early S, and that's going to be non-homologous end joining. DNA falls apart, DNA break occurs, the ends get chewed up, they, by some miracle, join up to the right ones again, and we're going to have mutations. Hall calls this illegitimate recombination, but on your boards they will call this non-homologous end joining, NHEJ, and it is an error-prone type of repair. You're going to need to know the difference between these two and how they work. Okay. So what are some of the lesions that we might see? I don't know if they've been putting this on the boards in recent times. I'll talk about it, but I'm talking about it mostly to get to this topic of multiply damaged sites. So there are these things called spurs produced by low LET radiation most often. They're produced by tracks, and it's where within a 4 nanometer area you have 3 ion pairs that are produced. You consider the DNA is 2 nanometers in width, so 3 ion pairs sitting on top of that can cause some damage. The blob is usually produced by high LET radiation. It's produced by tracks as well, but it's where there are 12 ion pairs in a 7 nanometer area, and I'll show you a picture in a moment. Most people believe that blobs produce multiply damaged sites. A multiply damaged site or complex lesion is defined by being a combination of damage that is 20 bases in size or in a 10 nanometer diameter volume of chromatin. It's produced usually by blobs, not by spurs. Here's the concept. And here's another name for it, is the clustered lesion. So sometimes it's called complex lesion, sometimes it's called clustered lesion, sometimes it's called multiply damaged site. All of them define what's going on here. So here is the spur. This is produced predominantly by low LET radiation. 3 hydroxyl radicals, 3 aqueous electrons in a 4 nanometer region. This sits on top of the DNA. It's not going to do that much damage. 3 hydroxyl radicals, it'll cause some breakage, but it won't cause a whole lot. But the blob, which is 7 nanometers in diameter, has 12 ion pairs. This sits on top of the DNA. And now we're going to get what we consider to be complex lesions or clustered lesions, where we have single strand breaks, double strand breaks, base damage, all within this same small 10 nanometer region of DNA. And why is this so bad for us? Because the cell doesn't know how to fix it. We've got to send in the machinery for single strand breaks, for double strand breaks, for base damage, maybe for crosslinks, and all this is in a very small region. You can't even fit that much repair machinery in there. So John Ward originally called it locally multiply damaged site because it's got multiple damage in a small local region. It's called cluster regions because it's got clusters of damage in a small region, and it's called complex lesions because it is a complex lesion made up of many different kinds of damages. All 3 terms are correct. I'm not sure. I think the multiply damaged sites is not as common on the boards these days as clustered lesions or complex damage. Okay, finally, old paradigm was that cluster lesions may be the result of a single electron track from low LET radiation. There is only a small amount of literature out there that shows this. It's very difficult to look for. But there have been at least 3 reports that with X-rays or gamma rays, one can get clustered lesions. Very, very rare, but it suggests that gamma rays, X-rays are more damaging than we would have thought. And again, just to kind of bring up how these multiply damage sites occur, this is where we have a lot of electron damage events, hydroxyl radicals, etc., all sitting in one area in a 20 angstrom or 20 nanometer, 20 length and 10 length of bases. I'm not going to talk about delta rays here. I've never seen on the board, so I'm going to just skip this. So this is the idea of the cluster lesion and how many there are. And I just want to point out that even no matter what anybody says, they are not that common a lesion. They're not that easy to detect. So here's that track. This is just to kind of give you a better idea of it. Here is an alpha particle track, and each dot represents a quantum of energy that's being released. This is actually coming from a track. They've superimposed a DNA helix on here, so you have an idea of what it looks like, but this is not real. The track is real, but the DNA helix isn't. And what you will see is from a lot of these tracks, there are side tracks that are coming out. And we see that much more with tracks from electrons than we see from alpha particle tracks. Here's a spur, here's a blob, and you can imagine this could be much more damaging than the spur is. Here is a short track coming off, here's a longer track coming off. So these tracks are capable of damage, and there is a random nature to it, as I keep pointing out, where, what hits the DNA where. So here are numbers you need to memorize. We haven't identified what a D0 dose is. We'll get that in our next lecture. But a D0 dose is approximately 1 to 2 gray. So you give mammalian cells 1 to 2 gray, and what kind of damage do you expect? You expect greater than 1,000 base damage lesions, 1,000 single strand breaks, and 40 double strand breaks. I would know those numbers. Here are the actual numbers for it that you can see. Damage bases, 1,000 to 2,000, single strand breaks, 1,000, double strand breaks, 40, and then lots of other things that you might see here as being important and worth taking a look at. But the fact is, those are the 3 numbers that I've seen them ask on the boards. Following, as I said, a D0 dose, they're calling it 1 gray here. It's not quite 1 gray. There are also some data on how many tracks actually go through people. I've never seen this on the boards. It's there for your information purposes. Like Penny Jago gave me this. Okay, a good friend of mine, Ray Mine, who passed away of cancer about a year ago, he gave me this slide, and I think it's extremely useful. And what we're trying to compare and contrast here is the damage that's caused by gamma rays and X-rays versus the damage that's caused by UV radiation. And there is a board's question that asks about this. For gamma rays, you can expect it's mostly going to be single base damage, a base gets changed. It's going to be single strand breaks, and it's going to be double strand breaks. For those not produced by ionizing radiation, but produced by UV radiation, you're going to have TT dimers, and you're going to have cyclobutylpyrimidine dimers. They're going to be repaired by an entirely different mechanism than the single strand breaks, double strand breaks, et cetera, et cetera. So that's worth remembering. Okay. So what we need to go talk about now is how do we measure these strand breaks? Some of the methods are very old, and I don't think they're going to be on your boards. Some of them are newer, and they will be on your boards. For all methods of measuring DNA strand breaks, however, I want you to remember that if you monitor over time, you are monitoring repair, right? So all these methods measure damage, but they also can measure repair by measuring their processing over time. The first method is an old method. I don't know that you need to know it in great detail, but I need to point out the alkaline versus neutral difference because that does come up on the boards and it's useful in a couple of different assays. So let's kind of look at this in perspective. What we're going to be talking about here is measuring damage to the DNA strand. This is not how the DNA exists in the cell, right? It exists in chromosomes. We're going to move up to chromosome injury yet, but we're going to start with how do we measure damage to the strand itself. We also know there's a superstructure to this where there are nucleosomes that function as beads on a string, and there are extended forms of different parts of the chromosome before we get up to full chromosome in size. And you can see many of these kinds of structures, the helix, the nucleosome, a solenoid, which is a twist of the helix, et cetera, until you get to the chromatid. Okay, so here's the filter elution assay, and I'll just mention to you what it does, but I don't think you need to know this in detail. What they used to do is they would give cells radiolabel, so the DNA becomes labeled. So you have radiolabeled DNA inside cells. You extract the DNA, you precipitate it with an acid called trichloroacetic acid, that doesn't matter. But one thing that you do need to know is that nitrocellulose filters have an affinity for DNA. So people would use nitrocellulose to bind to the DNA, it would be retained on the filter. They would then add a buffer which caused the DNA to come off or elute. And they did this, if they did it with an alkaline solution, then both the single strand and the double strand breaks would come off. If they did it with a neutral solution, only the double strand breaks would come off. So that meant that you could distinguish between single strand breaks versus double strand breaks. So first of all, alkaline elution, what did it do? If the bonds were a little bit labile, it broke them more. So you have single strand break, but it becomes very labile and it breaks now. So it would detect single strand breaks, period. But what are double strand breaks? In a way, double strand breaks are types of single strand breaks. They're just ones that are close to each other. So alkaline elution takes off single strand breaks, which include double strand breaks. But neutral situation was not enough to make the single strand breaks be labile. So a neutral solution, which could be anywhere from pH 9.6 up to 7, these would detect double strand breaks only. So this allowed for a measuring of the single strand breaks versus the double strand breaks, and therefore was extremely useful. Here are just some old data, fractions uneluted, the elution time. You can see neutral for normal cells, no radiation, 54 gray with neutral conditions, 54 gray with alkaline conditions, and you can see lots more breaks. What I want to point out is look at the doses here. These are whopping doses they have to use in order to detect the broken DNA. This was not a sensitive technique, and that's part of the reason why it fell apart. The DNA is associated with histones, and there are people that did sedimentation through sucrose gradients. That's never been on the boards. I'm not going to talk about it. The methods that are on the boards, there are going to be actually 3 that I'm going to talk about. The first is the COMET assay. So the COMET assay is a microgel electrophoresis system. It is very sensitive method, probably the most sensitive for measuring the damage to the DNA itself. Let me show a picture and then I'll come back to this. So here's the idea. This is a microscope slide right here. You put the DNA in the center. If the DNA is broken, and you apply an electric current. If the DNA is broken, it comes out and forms this comet. If the DNA is not, it stays here around the rim intact. You measure the amount of DNA in the comet tail, and you get a measurement of the amount of broken DNA. So this COMET assay worked really well. You can do the COMET in alkaline or neutral conditions. If you do it in alkaline conditions, you're going to pick up single strand breaks. If you do it in neutral conditions, you're going to pick up double strand breaks. Got to remember that difference between neutral and alkaline. It's been on the boards before. Now, the other thing I need to say is something about sensitivity. And if you do an alkaline assay where you're picking up single strand breaks, it is the most sensitive means for detecting the damage, not for detecting repair, but for detecting the damage. Follow it over time and you can detect repair. There's going to be another method we're going to talk about that gives us even better sensitivity. This is going to detect the damage to the cells. Okay, another method that is a probably more modern method, but it requires very high doses to detect it, is called pulse field gel electrophoresis. It picks up large fragments of DNA, it measures them for damage and repair, and it's usually where you do this sort of zigzag gradient. Let me explain it. This is a gel electrophoresis. The brightly fluorescent stuff is DNA. If the DNA is intact, it stays at the top. If the DNA is broken, it goes down to the bottom. These are different doses, 5 gray, 10 gray, 25 gray. So now what they're doing is they put this DNA on the gel, they run an electric current through it, but instead of running the current directly through where this is positive down here and this is negative up here, what they do is they zigzag it and change the direction of the current. And that makes the DNA sort of wiggle through the gel and allows for distinction of size. So you can see here that we're detecting most is broken at 100 gray. I suppose nobody's surprised by that. But we can start to see some breakage at 5 and 10 gray. But it also allows us to see whether there are particular DNAs that are breaking more than others. So this technique has become useful. There are some groups that use it mostly for detecting what kinds of DNA is broken. Here's another image of the COMET assay. The unirradiated cells, these were given a pretty whopping dose, 8 gray. You can see how big this comet is. And I should also mention that the programs that we use to measure the comet tail in space, we also use to measure the comet tail size here. There's another assay that I need to mention that was on the boards, I guess a year or two ago, and it's called the micronucleus assay. The micronucleus assay, it's simple. You look under a microscope and you look for a micronucleus. Micronucleus is just a small nucleus in the cell. What does the micronucleus contain? Most people believe that it contains an acentric fragment. We'll talk about acentric fragments as we talk about chromosomal injury, but here is the micronucleus. And so after cells have been irradiated, we count the number of micronuclei, and we get an idea of how much DNA damage there was to the chromosome, leading to acentric fragments. And we can even detect these by a kind of fish assay. Here's another picture of the micronucleus assay being used to examine chromosomal injury following radiation exposure. I will say it's a controversial assay. I think what you need to know is that it's used to measure chromosomal injury. It appears to be somewhat dose responsive in that you can see changes with dose, and it is therefore quite effective. Okay. Finally, we're going to get into chromosomal injury itself. And here we've got the normal cell. Here we've got the cell that's been irradiated. And we see here an acentric fragment and a dicentric. What is a dicentric? A dicentric means it has 2 centromeres. An acentric fragment means it's lost its centromere. When we're doing these assays, we're most often doing them on M-phase cells because we can see the chromosomes really well, whereas on interphase cells, we can barely see the chromosomes. We get the chromosomes, and in the old-fashioned ways, we used to line up the chromosomes and count them and look for abnormalities. Here's an electron micrograph to just show you how much protein there is on these chromosomes. So we're getting a lot of protein when we irradiate. We may even hit protein and not hit the DNA. So just because you hit the chromosome doesn't mean you're actually getting DNA damage. What are the parts of the chromosome? Here's the centromere or central body, or some people call it kinetochore. Here is the uh, telomere, which is at the very ends of the chromosomes. Here we have two different chromatids, and there are bands on these chromosomes that allow us to identify them a little bit better. Here's the short arm and the long arm. The short arm is often called the P arm. The long arm is called the Q arm. And it should be made clear that coding regions are not uniform across any chromosome. There are some regions that are gene poor and some regions that are gene rich. And you know, if the radiation happens to cause a damage here, it might not have any impact on the functioning of the cell. Whereas if it were to occur here where there are lots of gene products, that could cause significant effects. Here are the human chromosomes by convention lined up from 1 up to 22 and then Y and X with their different banding techniques associated with them. And you can see the structure. So there's a nomenclature for chromosomal abnormalities. I don't know that you need to know all of this, but for our purposes, it's important to know that a -1 means there's a total loss of chromosome number 1. A +7 means there's a gain of chromosome 7. A 4p plus means that there's additional material that's found on the p arm of 4. A translocation of 922 means that there's a translocation of these two chromosomes with a breakpoint in between. And then I think the other point to be made here is that about 1 micron has 25 million bases. So the DNA is very compact. It's all the information is very tightly packed within the cell. And here's just a smear from a cancer cell. You can see that there are many different abnormalities. This is an aneuploid cell line. It's got 4 copies of chromosome 2, 3 copies of chromosome 1. It's got a homogeneously staining region. It's got a bunch of marker chromosomes. So this cell is headed for trouble. And now let me bring up the difference between chromosome and chromatid aberrations. Just a moment ago, when we talked about the kinds of repair that go on, we said that it matters, the repair matters about whether there's a duplicate copy of the chromosome. Well, the kind of damage you get matters based on how many chromosomes and chromatids you have at any given phase. This puts in words what I'm going to try to show here in a picture. So here's the cell cycle. As I said, most cells out here are in G0, enter at G1, come to S, duplication of the genetic material, G2, and M. Well, if there are multiple copies, if the chromatids are actually separated, then we will get chromatid damage, which can happen late S or G2. If we don't have separate chromatids, we will get chromosomal damage, which is G0, G1, or early S. Now, which of those two is going to be more common? Chromosomal damage is going to be more common because most cells are in G0, G1, or S. But if this cell happens to be in late S or G2, we are going to get a chromatid injury. Now, you notice I keep leaving M phase out of this, and it's because there are some very recent literature that claim, and I mean like in the last year, that claim that there is virtually no repair during M phase, which would mean there's no way of rejoining broken chromosomes. There's no way of being able to measure damage. So I think the general consensus right now would be that if anything happens during M phase, it doesn't get repaired at all. I'm not 100% sure I agree with that, but that's the convention. Okay, so let's go back and look at what we've said here. Most cells reside in G0, G1. We've said that already. If we want to be able to detect them in M phase, we need to give them some sort of mitogen to stimulate them. Phytohemagglutinin is a commonly used one. G1, there's a single chromatin strand. In G2M, there are the 2 chromatin strands. And therefore, from interphase damage, we're going to get chromosome aberrations. else other times late SG2, we're going to get chromatid aberrations. Says the same thing. Okay. So back in grade school or high school or something, you learned about anaphase and prophase and telophase. And these you may not have been as well aware of it then, but these were phases of M phase. And the reason why they had all these names with them is because you could observe them under the light microscope and see what was going on. One thing that probably you will recall was that the centromere played a very critical role in this. If, as the cells lined up at the equatorial plate, they would line up with their centromeres next to each other, and the spindle fibers would grab onto the centromeres or the kinetochores or whatever you call them and pull them into daughter cells. So if a chromosome has a centromere, it gets pulled into the daughter cells. If it doesn't get pulled in the daughter cells. And this is going to be important to us here. Here's just a pretty picture to show you this event taking place. And here's the diagram to show the importance of the centromeres who are being grabbed onto and pulled into the daughter cells. So here we go. There are lots of lethal aberrations in cells when they've been irradiated, but there are 3 you need to know for your boards. Number 1, most important, most critical, most common is the dicentric. What happens in the dicentric? 2 breaks in 2 chromosomes. They join together inappropriately. One has 2 centromeres, and actually this is missing up here. There should be a centromere right here. One has 2 centromeres, therefore it's called a dicentric because it has 2. And one has no centromeres, and therefore it's called acentric. Okay, so think about this for a moment. Remember, we just said a moment ago, acentrics are what make up those micronuclei that we're looking at microscopically. So that's one way of being able to measure them, or you can look at them here. The fact is, when a cell has an acentric fragment, what's going to happen to it? The dicentric, the centromeres will bind, they'll get pulled into the daughter cells, no problem. But when you've got an acentric fragment, what's going to happen to that piece? It's going to be lost. It can't bind to a spindle fiber. It's not going to be pulled into daughter cells, and it's going to be lost during cell division. It might take some time to lose it, but it will be lost. That is usually a big chunk of DNA. The cell can't survive to lose that much DNA in the cell. So the bottom line is, see an acentric fragment in the cell, that cell's destined to die. It might take months to die, but it's going to die. Second most common lesion is the ring. This is where you have 2 damage events on a single chromosome. They come together and form a ring and a large acentric fragment. This goes to divide, acentric fragment is lost. So having a ring is also a sign that that cell will die, a lethal. Why is the dicentric more common than the ring? Because 2 hits in 2 chromosomes is more likely than 2 hits in the same chromosome. Finally, the third lethal aberration is a chromatid aberration. It's called an anaphase bridge because it looks like a bridge during anaphase. We'll see one in a moment. Here we go, 2 hits. These guys join together and we get a bridge, an anaphase with an acentric fragment. This acentric is lost. This cell is destined to die as well. I'm not going to use, forget this. We'll talk about triradials later and quadradials later. They'll be important, but here's some pictures. Here's the dicentric, acentric fragment. Here's the ring. Here's the anaphase bridge. All these cells, all 3 of those cells that have those 3 aberrations are going to die. It might take months, but they will die. Okay. There are aberrations that we call kind of symmetrical aberrations. There are ones that are called asymmetrical. The ones we talked about, all of them are asymmetrical because we end up losing a chunk of DNA. If it's symmetrical, you retain all of your original DNA. It might not be in the same place, but you retain it. If it's asymmetrical, you've lost it. I'm going to show a better diagram in a moment. So here are the lethals, the dicentric, the ring, and the anaphase bridge. We're going to talk about this alpha-beta ratio stuff in probably next time, but it's there for you. And here's just the summary of the chromosome versus chromatid aberrations that might be useful. But now we're going to talk about I'll talk about 3 non-lethal aberrations. 1, the symmetrical translocation. You're used to this, you remember this from cancer studies in medical school. Cut in 2 chromosomes, so just like the formation of the dicentric, except these guys join up to each other. So they each retain their centromere, but they have some DNA that's been exchanged. Most of the time, there's a little bit of change around the break point, but it's considered a symmetrical exchange because there's not that much loss or gain of DNA. Number 2, 2 hits in the same chromosome, kind of like the ring, except instead of joining up with the end, this joins up with each other, and this piece gets kicked out. In my opinion, this looks kind of big. It should be a lot smaller than this. This is usually called the small deletion because it's got a small loss. I guess they made this big to make it be more obvious. So how are these going to be relevant to us? Symmetrical translocation. We have lots of symmetrical translocations in the population. In fact, some of us may have symmetrical translocations. There's nothing dangerous with them. Occasionally, they give rise to oncogene activation if they hit oncogenes as they're rearranging. But there are people that have lived with symmetrical translocations their entire lives for 40, 50 years and never had a problem with it. There is a high background of symmetrical translocations in the general population. However, small deletions how are they going to be damaging? If there's a tumor suppressor gene on that region, then we're going to put the cells a step toward a cancer. So the symmetrical, it's going to be oncogene activation. The small deletion, it's going to be loss of a tumor suppressor gene. So here are just a few of these translocations that we all are familiar with a exchange DNA. This is a 2,5 translocation. Here are a bunch of translocations that have been found associated with different events, different kinds of malignancies. They're easy to detect in lymphoid cells, so we pick them up pretty easily there, but you can pick them up in other kinds of tumors. There is a third rearrangement that's not in Hall's textbook that I want to mention to you because it is another symmetrical translocation and it's been on the boards. It's called an inversion. It's very simple. You get 2 cuts in the same chromosome and then the DNA flips over and you end up getting them joining up with the wrong ends. It's pretty much the same as a translocation, except for both rearrangements taking place on 2 chromosomes, they take place on 1 chromosome. It's symmetric, and it is not lethal because you haven't lost that much DNA. So the translocation and inversion are symmetrical. The interstitial deletion, the dicentric, and the ring are all asymmetric chromosomal aberrations. The translocation, inversion, and interstitial deletion are all non-lethal. The dicentric, the ring, and also the chromatid aberration, the anaphase bridge, is lethal. There are other lesions that we'll talk about along stream. This just shows how these are formed. I don't think you need to know that, but there is a triradial that we'll talk about with this characteristic of particular disease called Fanconi's anemia. And this just shows some of these as well. This shows the acentric ring that forms the micronucleus. Okay, let me see where I can reach a stopping. Okay, I'll do this one, and then we'll go, and then we'll stop. Okay, so there is a radiation accident, and Fukushima happened, and the symmetry was pretty poor immediately after the accident. So one of the things people often try to do is to use biological markers to look for what the dose could have been. And chromosome aberrations are frequently used because chromosome aberrations are directly related to dose. So the question is, there's an accident that takes place, what would be the best thing you would use? Well, if you are looking at the first 6 months following the accident, dicentrics are the best. There's no natural background of dicentrics. Those cells will die within 6 months. They're present in the largest quantity. So dicentrics would give you a good marker. Dicentrics usually have a sensitivity down to about 25 centigrade, and they're totally dose responsive. But now, it's almost 10 years after Fukushima. Are we going to see any dicentrics in those people? Nope, we're not going to see them. So what would we try to use today? It would be, we have to use great caution. We would try to use translocations. We'd have to do a large amount of work in the background population to know what the rate of translocations was in that population, but translocations would be the best thing to give us some idea. So that's how we use these as biological dosimeters. Now, I think I've told you this before, but there's a question in the pool of boards questions that says, a patient has just been transported to you from a neighboring hospital and has gotten a radiation dose for treatment, and you don't know what the dose is. Which of the following tests would you use to check dose? And they give, rings and dicentrics and micronuclei and everything. And the answer is not any of those things. It's call the physicist up at the other hospital and find out what they gave them, okay? Because these are only good as estimates during accident situations where we don't know dose, but our physics gives us the best answer. Physics is always trumps everything when it comes to looking for dose. And I think this is a good, I'll summarize the aberrations next time before we start again, just to put us all on the same page. And we'll get into some of the methods we use for measuring chromosome aberration. Okay, so I think I got through this slide last time, but I want to go back to it and recapture where we were. So if you will remember, what we were talking about was these development of chromosomal aberrations. And the idea is that if we look at the process, we've got ionizations. They can be indirect or direct. Ionizations that can lead to direct or indirect damage. These can be completely repaired. The cell goes on, there's nothing happens. This especially happens a lot at low doses. But the damage is processed. It can be, if there is residual damage, among the things that can happen are things like point mutations. But chromosome aberrations are what we talked about last time. And these chromosome aberrations can be lethal, as we see in dicentrics, rings, and anaphase bridge. Or actually, anaphase bridge is a chromatid aberration, but it fits in that category. Or it can be a translocation or an inversion, reciprocal translocation and inversion, or small deletion, in which case the cell is able to survive, often with effects on it. However, sometimes cancerous, sometimes it's significantly mutated. It just depends on the issues. So I was going to summarize these aberrations. They're the chromosomal aberrations that we talked about before. The cells are radiated in G0, G1, early S before duplication of the chromosomes. Usually there's a break that occurs. There can be translocations, which are not lethal, and dicentrics and rings, which are both lethal. And as we said last time, in the case of a radiation accident, if we don't really know what the dose is, or if we need to guesstimate dose, in the first 6, 8, 9 months after the accident, we can use dicentrics as a monitor. That sensitivity is 25 centigrade, but dicentrics are lethal. They're gonna die in the patient's cells within probably the first 6 months or so. So beyond that, we would have to use translocations. Translocations are extremely complicated to be able to do because there is a background rate of translocations in any population, and that needs to be determined, and that can vary from one area to another. Chromatid aberrations are when the cells are radiated after duplication of the genetic material, late S, early G2. There's a break on both chromatids that leads to an anaphase bridge, which is lethal. And that is the only chromatid aberration that we talked about. There are some others, but the only one I've seen relevant to the boards is the anaphase bridge. there is a correlation of chromosome aberrations with physical abnormalities, mental retardation, cancer, and also in some cases lethality. Now, I want to make one point here that I'll probably make later, and it's about this issue of mental retardation. We haven't yet carefully defined what are stochastic and what are deterministic effects of radiation. A stochastic effect is one that happens by chance. It's rather random. It can occur anywhere at all. It usually involves a single cell effect. And there are deterministic effects that usually are what we would consider tissue effects. In fact, in EU, they define them, they don't call deterministic effects anymore, they call it tissue effects. And the only point I want to bring up is that mental retardation can be either stochastic or deterministic. And let me explain the two different distinctions. If mental retardation comes about as a result of a chromosome abnormality in the eggs or sperm that are passed on to the offspring, which is in part what we're talking about here, that would be stochastic. There's a genetic abnormality that is passed on. It is retained in the genome. If, on the other hand, as we'll talk about with the Japanese atomic bomb survivors, mental retardation happens after an in utero exposure, then it is considered to be deterministic or a tissue effect because it doesn't have the genetic component. It has a component of if you damage enough cells, then you see an effect. So mental retardation can be either stochastic or deterministic. We will talk about that again later, but I wanted to bring it up now because this was defining the stochastic mechanism. Okay, there are several assays that we use for detecting chromosomal aberrations. We looked a lot at smears and how people will line up the chromosomes and detect them. But there are some more modern methods, and the modern methods are frequently asked on the boards. FISH, fluorescence in situ hybridization, is shown here. And what's done there is a small probe that is specific for a particular gene or gene region is labeled with a fluorescent probe and is hybridized to the chromosome. And wherever they turn out to have matches, you see this fluorescent dot appear. In this particular example, a sequence that was specific for centromeres was made brown here. A portion that was specific for telomeres was made white here. And a region that was specific for this particular chromosome was made yellow here. And so this method is called fluorescence in situ hybridization because you're using fluorescence, you hybridize the chromosome all by itself in situ, and it is a hybridization process. Now, a more common technique, but it is more difficult, it's trickier, is a method that's called spectral karyotyping or SKY. It's sometimes called chromosome painting. And it's where you make a different colored fluorescent probe that is specific for each chromosome. You can then stain every chromosome, see this multicolored image, and immediately claim which ones are rearranged and which ones aren't. Here is, for instance, a spectral karyotype from a cancer cell that has extra copies of several chromosomes and some rearrangements. And you can go back to a guide that's color-coded for the different chromosomes and say exactly what those rearrangements are, often picking up things that wouldn't have been detected in a straight just chromosome smear that we're used to. This is an example of spectral karyotyping or chromosome painting done in Mike Cornforth's lab a number of years ago. Here's what the smear would look like without chromosome painting. Here it is with painting, and here you can see what it actually looks like. Rearrangement right here that maybe you would have missed otherwise, a dicentric and acentric fragment, and very easy to detect abnormalities. This is another similar example of chromosome painting or spectral karyotyping here, done by Tej Pandita, a friend of mine. And what this is the untreated cells. These are cells that have been treated with 1-gray. And you can see here, for instance, is a rearrangement that, again, you would have picked up maybe in standard karyotyping, but it would have been very difficult. So this technique Fish, this is a type of fish. Spectral karyotyping is a type of fish that is specifically designed to see each chromosome, whereas gene-specific fish is the kind that we showed first. So on the boards, if they ask about fish or spectral karyotyping or chromosome painting, you will know what it is, and most often they're using it for the definition, so you have to know what it works with. And here's one other example. This is in Hall's textbook showing a dicentric with a rearranged fragment and a translocation here. Okay, now we're going to get into a concept that's going to begin here and carry over into the next topic, and it relates dose to, in this example, chromosomal abnormalities. In our next example, we'll relate it to cell survival and cell killing. So here's a plot. You know, British biologists got to plot everything, look to quantitate number of aberrations, and by this they mean dicentrics, the number of dicentrics per cell against the absorbed dose. So what they're doing is they're treating cells with higher dose, and then they're counting the number of dicentrics they find per cell. What they predicted was that they would see a curve that was either linear or a curve that was quadratic. Unfortunately, what they saw was a combination. The lower part of the curve was linear, the upper part of the curve was quadratic. Because of that, this is often called the linear quadratic model, because it's a combination of linear and quadratic. What does that mean? A linear relationship means that there is a direct relationship between dose and your endpoint, in this case, dicentrics. A quadratic relationship would mean that there is a relationship between number of aberrations and dose squared. So these look very different on a curve. The linear model looks like a straight line. The quadratic looks like an asymptotic line that's reaching upwards. And so what people said was, look, if you look at the actual results here, there's a low dose portion that is linear, a high dose portion that's quadratic. How could we possibly get this? At low doses, think about it for a moment, oftentimes there's one track or less going through each cell. So the only way to get a dicentric is if a single track damages multiple different chromosomes. Whereas at high doses, where there are lots of tracks going through each cell, now we can have multiple tracks causing multiple chromosome damages, which give rise to dicentrics. So the chances of getting a dicentric at high doses are much higher, but it is related to dose squared because of this multi-event that is occurring. The fact that multiple tracks are coming through, not just a single track. So the model said, and continues to say to this day, at low doses, there's a linear relationship with dose where a single track causes multiple hits in the chromosome, which now give rise to a dicentric. In some rare examples, very rare, the number here that we're getting of aberrations per cell is pretty low. At high doses, it is much more common to be able to get dicentrics. And it could be happening by this linear method, but more than likely, it's way overwhelmed by the number that are happening by this method, where you're having multiple hits that occur, and these are giving rise to the dicentrics. If we then put relationship mathematics on this, the idea would be that the yield of dicentrics would equal this, which is a constant called alpha times the dose, plus this, which is a constant called beta times the dose squared. So this gave rise to the idea of what we call the linear quadratic model, and the so-called LQ model is still the most common and most scientifically useful model. There are a lot of limitations to it, and I myself have written articles about the limitations of the linear quadratic model and its applications, but the fact is this model for sure holds up with regard to dicentrics. Does this make sense, or do I need to say it again? Okay, I'm not hearing anybody say anything, so I will keep going. So eventually what we're going to do is we're going to, in our next topic, take these aberrations we detect and relate it to the survival curves that we usually work with. Now, I want to point out that in this situation, we were looking at mostly dicentrics, but you could actually look at translocations as well, which would maybe be more meaningful, but nobody does it. Why are they looking at dicentrics? Because dicentrics relate to cell killing, and they're trying to relate this to a survival curve. If we were looking at translocations, it would relate much more toward cancer, and therefore would probably give us a different measure for being able to look at this. Okay. We've talked about a lot of methods for being able to detect strand breaks within cells. We talked last time about alkaline versus neutral elution, comet assay, pulse field gel electrophoresis. We moved into chromosomal abnormalities. Now I want to mention one that I will come back to again in this course, but it is one that is very, very commonly used in the literature, and if it is not on your boards, I will be shocked beyond measure. And it is called the gamma H2AX assay, or the assay, and this should have a gamma in front of it, gamma H2AX foci. What does this measure? What are we looking at? Gamma H2AX is a histone protein, H2AX, that is phosphorylated, that upon irradiation is phosphorylated not in the alpha position, not in the beta position, but in the gamma position. So the presence of gamma H2AX means that cells have been irradiated and are going to repair. The absence of gamma H2AX means that we still have lots of H2AX in the cells, but the cells have not been damaged in any way. So this has become a marker for damage, even though gamma H2AX is actually a marker for repair. But repair and damage go hand in hand. So one way of being able to monitor whether cells have been damaged is to look for the repair machinery. This was done by Bill Bonner, a friend of mine, and what he did in these studies is he looked at cells that were unirradiated, irradiated for 0.6 gray for 3 minutes, 15 minutes, 30 minutes, 60 minutes, and 180 minutes, and looked for these little spots within the cell called foci. What these are is he's staining with a fluorescent antibody against gamma H2AX. There are none in the resting cells, but when you irradiate, you start to see these spots pop up. And here at 15 minutes, you can see these beautiful foci that are formed. These foci are believed to be locations within the DNA where repair is taking place. So gamma H2AX has been recruited there, and repair is taking place. And these persist for up to 3 hours after radiation exposure, even longer, we'll learn later. So this assay, the gamma H2AX assay, is not per se a measure of DNA damage, but it is a reflection of damage because it's measuring repair. And it has become the most sensitive assay for detecting whether damage has taken place. It's even more sensitive than the common assay in many examples. It also persists for a period of time, but it's never going to be useful in an accident because it will disappear after hours after exposure. So it's not going to be useful in an accident, but it might tell you something about the cells you're working with and what the extent of the damage is. As I said, we will talk about this again later. So here's just sort of a summary of some of these methods that we talked about. This might be useful when you're coming for studying. We mentioned sucrose gradient centrifugation, but we didn't really talk about it. Here's the filter elution assay we talked about before with the alkaline and the neutral detecting mostly double strand breaks, whereas the alkaline detecting single strand breaks, pulse field gel electrophoresis, which doesn't have a very good sensitivity. We didn't talk about nucleotide sedimentation. I don't think you need to know it. The comet assay we talked about as well, alkaline and neutral. FISH we just mentioned as a method for being able to detect whether the cells are have had chromosomal aberrations. We did not talk about premature chromosome condensation and I haven't seen that on the boards, so we won't talk about that either. And then finally, there's gamma H2AX, which we just mentioned, and has a really great sensitivity. Okay, so we're going to move into cell survival curves. That will be our topic next, but I want to cover a few things here kind of as introductory material. This is the log of the surviving fraction against radiation dose. And here is where diploid cells sit, tetraploid, hexaploid, and haploid cells. Why are haploid cells the most radiosensitive of all these, have the lowest, have the most sensitivity? Because there's only one copy of each repair gene, and if you damage it, the cell can't really replace it, and so it dies. Diploid is better because there are two copies, damage one, the other one's still there. Why are hexaploid and tetraploid so difficult? Well, mostly because the DNA there's much more DNA per cell, and it becomes a larger target to be able to hit. This is going to be the standard survival curve that we're going to talk about in great detail next time. There are many consequences of radiation exposure. We will talk about these. Reproductive failure is going to be the one that we consider to be most important. One final point I want to make here that is not in Hall's textbook yet, but I think it helps in how we think about radiosensitivity and radioresistance in cells. So Burgundy and Tribundeau proposed a law, or what they called a law, many years ago. And that was that you could determine radiosensitivity versus radioresistance of cells based on two criteria. Number one, are they differentiated? Number two, are they dividing? And the idea was if they are dividing, but they are not differentiated, they will be very radiosensitive. And if they are differentiated and not dividing, they will be very radioresistant. And indeed, this is the case for most cells in the body. They categorize these into vegetative intermitotic cells that divide, have no differentiation between divisions, most radiosensitive. These are going to be stem cells. And we're going to see a chart where different cell types fit into these categories. There are differentiating intermitotic cells that divide all the time, differentiate only to some extent between divisions, and they're pretty radiosensitive too. There are reverting postmitotic cells which don't divide. They are capable of division if you give them the right stimulus. I'm thinking here about liver, where liver doesn't usually divide very much, but injure the liver and it starts to divide. These are variably differentiated. They're pretty radio resistant. And then finally, the fixed postmitotic cells that are not dividing, are highly differentiated, are very radio resistant. And these would be some of the neurons, for instance. I wanted to make you aware of this because as we think about cells in the body, as this progresses, I think that this frame helps us. I will say now that the one thing they ask on the boards frequently is the exception to this rule. The one exception to this rule, we'll talk about a little bit later, is our lymphocytes. Lymphocytes are actually reverting postmitotic cells. They sit in our bloodstream. They're G0 stage. They normally don't divide until they encounter antigen, yet they are the most radiosensitive cells in the body. B cells are, for instance, extremely radiosensitive down to 80 centigrade. So that's going to be the exception, and it's frequently asked on the boards. Okay, there's a section on DNA repair here that I am not going to talk about now. I don't like Hall's description. I don't think what he has is sufficient for your boards. We will talk about a whole broad topic on DNA repair a little bit later in this course. So let me summarize the chromosomal story. So there are these chromosomal and chromatid aberrations that can be lethal, particularly if it involves the loss of a centromere. The chromosomal aberrations that are lethal would include rings dicentrics, and the chromatid aberration would be the anaphase bridge. The ones that are not usually lethal are reciprocal translocations, small deletions, and I would add to that inversions. And generally, we can use chromosome injury to give us an estimate of dose when a dose is not known, but when a dose is known, we should use our physics to tell us the answers.
is in the air for you and me.